Everybody. I'm Anu Budu. I'm the program manager at the Liver Cancer Program, and I want to welcome everyone this morning. Thanks for joining our seminar series today. We're very happy to welcome Dr. Shalev Itzkovitz from the Weizmann Institute, and we're looking forward to his presentation. Li-Shun Ma is going to formally introduce Dr. Itzkovitz, but I just wanted to note that we have an upcoming seminar on Monday, June 21st. At 10 a.m. Eastern Standard Time. Jesper Anderson from the University of Copenhagen will be presenting on oncogenomics in cholangiocarcinoma. So we hope you can join that as well. For today's seminar, if you have any questions for Dr. Itzkovitz, please add them to the chat box, or you'll have a moment after the seminar, just turn your mic on to ask your question. So thank you for joining us today, Dr. Itzkovitz, and Li-Shun, go ahead.
Thanks, Anu. And my name is Li-Shun Ma. I'm a systems biologist with a special interest in single-cell analysis in liver cancer. So today it's my great pleasure to introduce Dr. Shalev Itzkovitz. Dr. Itzkovitz uses tools from systems biology to study design principles of tissue organization, focusing on key metabolic tissues, the liver, intestine, and pancreas. He obtained his bachelor's degree in physics and mathematics at the Hebrew University in Jerusalem, master's degree in electrical engineering at the Technion, and a PhD in systems biology at the Weizmann Institute. Following a postdoc training at MIT, Dr. Itzkovitz joined the Department of Molecular Cell Biology at the Weizmann Institute in 2012 and obtained tenure in 2017. He has published over 100 research articles in peer-reviewed journals, such as Science, Nature, Cell, Nature Biotechnology, Nature Metabolism, etc. He was selected as an HHMI International Research Scholar and a Valley Young Investigator in 2017, and was awarded both daughter and consolidator ERC grants for his research on the mammalian liver. Without further ado, let's welcome Dr. Shalev Itzkovitz.
Okay, thank you very much. Thank you, Li-Shun, thank you, Anu, for the kind invitation. Today, I'll tell you about some of our work on spatial heterogeneity in the mammalian liver. As Li-Shun said, we're really interested in understanding how single cells work together within the context of mammalian tissues to collectively achieve physiological goals. And we work on the gut, the liver, and the pancreas, but all this talk today would be on the liver. As all of you know, the liver is the metabolic hub of our body. It stores all the nutrients after we eat and then releases them in a regulated manner in between meals. It detoxifies blood, produces albumin, bile acid, cholesterol, most of the carrier proteins, complement system proteins, really a very diverse set of functions, all performed by a single cell type, the hepatocyte. But the liver is very far from just being a bag of hepatocytes that all autonomously and equally perform all of these functions. It is actually highly heterogeneous, and this heterogeneity is related to the spatial structure of the liver. So the liver, as many of you know, is composed of repeating anatomical units called globules. These are the hexagonal shaped columns. They're about 0.5 millimeter in diameter in mice, about 1 millimeter in diameter in human. And blood flows from the corners of the lobule inwards through the radial sinusoidal blood vessels and drains out of the central vein. So in this blow up, the blood enters from the portal node, flows inwards and drains from the central vein. And this architecture makes it important for the hepatocytes not to act autonomously. I'll just give you one example. We know that oxygen is limiting for the liver. Only about 20-30% of the perfusion is from arterial oxygenated blood. So if the hepatocytes in the outer layers of the lobule would respire a lot, they would take a lot of oxygen, they would have a lot of available ATP for their energetically expensive tasks. But as the blood would flow inwards, oxygen concentrations would reduce to dangerous levels, suffocating the inner hepatocytes. And by measuring oxygen levels along this radial lobular axis, people have shown that it goes down, but it remains safely above hypoxia. And so this just means that the portal outer hepatocytes, they basically adapt their functions to make sure that the tissue as a whole will function optimally. Now, so this is the architecture: polarized blood flow, creating gradients of oxygen, nutrients, hormones. There's other morphogens that are also graded, and in response to this structure, when you just section the liver and start staining for different properties, you immediately see that the liver is highly zonated. This is just an example here of albumin, sort of a known hepatocyte marker. And you can see that the hepatocytes in the portal layers have much higher RNA levels of albumin compared to the hepatocytes in the central layers. All of the hepatocytes have high levels of albumin, but there's about 2 to 3-fold decline in the expression from the portal to the central layers. Glycogen content is also zonated, and a few dozens of key hepatocyte functions have been shown to be zonated for a very long time, for about 40 years. This is a phenomenon that's known as metabolic zonation. And in my lab, we wanted to get a global view of the zonation of hepatocytes along this lobule radial axis to understand the spatial division of labor of all of the hepatocyte functions. And so to start to address this challenge, a few years ago, my lab has started to apply a technique called single molecule FISH, single molecule fluorescence in situ hybridization, which enables us to take tissue sections and visualize individual RNA molecules as diffraction limited spots under a fluorescent fluorescent microscope. So here, every red dot is a single RNA molecule of beta-actin in the liver tissue. The blue are the DAPI-stained nuclei. And the way we get this very high signal-to-noise ratio is that we design libraries of probes, short 20 base per DNA sequences that we design to be complementary to sequential parts of our transcript of interest. So we get the plates with these probes, we pull all of them and we couple all the probes to the same fluorescent molecule. And now we take tissue sections, we fix the tissue, permeabilize them and hybridize them with the probe libraries. The probes are very small, so they readily enter into the cell and they all localize on our transcript of interest. So our RNA molecule, which is about 50 nanometer in diameter, now becomes a very bright spot with about 50 fluorescent molecules that are specifically accumulated on it due to the DNA RNA hybridization. And the trick here is that we use many, many short probes, unlike traditional in situ hybridization that uses a long probe that's stochastically labeled with fluorophores. When you have a long probe, any sub-sequence could bind an off-target. There are a lot of off-target in the cell, and then you get all the fluorophores on the off-target. Here, any probe could stochastically bind an off-target, but the probability that more than one probe would bind the same off-target becomes exponentially small. So the background is like 1 or 2 fluorophores, the signal is 50 fluorophores, and we can actually identify individual RNA molecules, count them, and get the digital gene expression while maintaining the structure. And we can do this for the entire lobule. So this is a part of a lobule. That's the portal node here. Central vein, the green are the membranes, the blue are the DAPI-stained nuclei. So the blood flows from the portal node inwards, drains out of the central vein. And every red dot here in this image is a single RNA molecule of PepsiK1, a very important liver gene, a hepatocyte gene, sort of a rate-limiting enzyme in the process of gluconeogenesis, the process by which the liver produces glucose to our body after all the internal glycogen stores have been degraded. For example, when we wake up in the morning, the liver continues to supply glucose to our body because our brain runs on glucose, but all the glycogen stores have already disappeared. And so the hepatocytes basically, they take up material and they build through a very ATP demanding task, build up glucose from 2 or 3 carbon sources like lactate, glycerol, amino acids. And you can immediately appreciate that there's a profound heterogeneity. There are hepatocytes with a lot of RNA molecules and hepatocytes with lower levels of expression, but there's also a spatial structure, and you can appreciate that when you count the dots. We have software that counts the dot in these images, and we sort of make Google maps of expression here and basically tell the computer that we want to calculate the RNA concentration in different lobule layers as a function of the distance from the central vein. So we count the number of RNA molecules and divide by the volumes of these layers. And this gives us RNA concentration of PepsiK1 versus the distance from the central vein. So this is the zonation profile of PepsiK1. And you can see that expression is higher in the portal regions. This makes sense. This is the region where oxygen comes in. So there is more ATP availability to carry on this synergetically demanding task. So this is one example, but of course, we don't want only one gene. We want to get the zonation profiles of all the 30,000 genes in the genome, or all the 8,000 genes expressed in hepatocytes. And of course, achieving this with a single molecule FISH is infeasible, right? So we calculated we would need something like 300 PhD students in order to design an image these 8,000 libraries, and we didn't have this kind of these kind of resources at the time, also now. But so we decided to take on a completely different approach to get a global zonation map of hepatocyte gene expression. And the approach we took was to perform single-cell RNA sequencing. So now it's a completely orthogonal approach. We teamed up with Ido Amit from our building here from the immunology department at Weizmann. We basically dissociated the liver and sorted thousands of hepatocytes and performed single-cell RNA sequencing, giving us these matrices, these tables of 30,000 genes by 1,500 hepatocytes, where now for every hepatocyte, every column is a hepatocyte, and for every hepatocyte, we get not only the expression of one gene, but the complete transcriptome. So this is great. But of course, the problem is that once we dissociate the tissue, we completely lose track of where each hepatocyte came from. So we don't know the zone, the original zone of each hepatocyte. So to retrieve the zone, we went back to the old literature on zonation, and we designed single molecule fish probe libraries for known zonated genes, such as this glutamine synthetase here that you can see is GLUL, only expressed around the central vein, or CYP2E1, the enzyme that breaks down the paracetamol. You can see it's expressed in the gradient from the central vein outwards. Albumin, you already saw before, has an inverse gradient, high in the portal regions, low in the central regions. And for each one of these genes, we segmented many hepatocytes, recording their layer, their position along the portal central axis, and counted the number of RNA molecules and sort of created this digital gene expression signature. So now you give me an hepatocyte and you tell me that it has 300 copies of this gene ASS1, and I can say what's the probability that this hepatocyte came from the pericentral layer 1 or layer 2 or layer 3, etc. We divided the structure into 9 concentric layers. So now we have these landmark genes. We call these landmark genes. These are genes that the expression of which reports on the location of the hepatocyte along the lobule radial axis. So now we go back to the single-cell RNA sequencing, and we don't look at all the 30,000 genes in the hepatocytes, but only at the landmark genes, the genes we mapped under the microscope. For example, this periportal blue gene, the pericentral red gene. And now, for example, if I have an hepatocyte with 34 copies of the red gene, 2 copies of the blue gene, I can infer it must have originated from the pericentral inner lobule layers. If I have another hepatocyte with 4 copies of red, 50 copies of blue, I can infer it must have originated from the outer layers. And this way we can build an algorithm that basically classifies each one of these 1,500 hepatocytes into these 9 concentric layers, and then we can group together hepatocytes. We have several hundreds of hepatocytes from each layer. We can group them together and computationally average the expression of these single cells to get a genome-wide, a transcriptome-wide zonation map of hepatocytes. So now we can go to any gene we've never measured under the microscope and simply average its expression in these spatially stratified, computationally stratified hepatocytes. Okay, so that's the idea. This gives us the complete zonation profiles of all the hepatocytes. Of course, it's critical to validate that this actually works. And the way we validate it is using single molecule fish. So basically what you're seeing here is zonation profiles, expression versus the layers from layer 1, which is the pericentral layer, to layer 9, the periportal layer. And the blue is the prediction based on the stratified single-cell RNA sequencing. And red is the expression as we measured using microscopy, as we measured for PepsiK1 that you saw in the first slides. And you can see that we have quite a nice concordance between these 2 measurement techniques. And so we believe the zonation table we're getting out of the single-cell reconstruction. So what were the results? The results were quite surprising for us. The previous estimates on the number of zonated hepatocyte genes along the mouse lobule radial axis were about a few hundred, 300. And we found that actually about half of the hepatocyte genes are zonated. There's about 7,000 genes expressed in an hepatocyte, in a mouse hepatocyte. 3,500 of them are zonated. These are all the zonated genes. So the rows here are genes and the columns are the lobule layers. So we have pericentral genes, we have periportal zonated hepatocyte genes. And we also found quite a few non-monotonic genes that actually peaked at the mid-lobule, like this hump over here. And this was interesting because the mid-lobule region, the mid-lobule zones were considered the transition between portal genes and central genes, but we actually found that there are some functions that are distinctly allocated to these mid-lobule layers. This is an example of a hump encoding hepcidin, a very important hormone that preserves iron homeostasis. And you can see that the expression, every bright dot is a single RNA of this hump. Expression is low around the portal node, then it peaks around the mid-lobule, and then it goes down towards the central vein. And this is very interesting because you can now start to ask questions how the tissue generates this kind of non-monotonic profile. It could be an inhibitor from the central vein and another inhibitor from the portal vein, or maybe a short range inhibitor and a long range activator. And since you have a global map, you can start testing hypothesis about the morphogenetic gradients that could give rise to these kind of interesting profiles. Now we can, since we have a global map, we can start looking at specific functions and ask about design principles. Which functions are allocated periportally and which are pericentral? This is an example of genes involved in liver secretions. We know the hepatocytes transcribe genes that encode proteins that are secreted, such as carrier molecules like albumin, complement system, clotting factors. And most of these genes are expressed periportally, similar to albumin that you can see here in this single molecule fish validation. Again, this makes sense. Most of the ATP of a cell goes into translating proteins. So translation is a demanding, ATP demanding task. And it makes sense to allocate the translation of these secreted proteins to the layers where there's more oxygen availability. Xenobiotic metabolism, the detoxification of foreign material. Most of the enzymes are pericentral, like this TYP2E1 you see here, but there's also some interesting exceptions, like you can see here for GSTA2. And there is an interesting story for each one of these. We can generally group together different functions and examine their average zonation, but we can also do more than that. We can also examine computationally the entire metabolic map of the hepatocyte. So here in this metabolic map, every node is an enzyme, and these are basically metabolic cascades that the hepatocyte is applying. And what we wanted to ask is whether there are enzymatic cascades where sequential enzymes in a metabolic cascade are expressed in sequential lobule layers. So maybe the hepatocytes as a whole can sort of distribute the enzymatic cascades over several sequential hepatocytes. And so I want to show you a few examples of these kind of production line processes. So one interesting example is this system here, where we have an enzyme glucose-6 phosphatase that basically takes glucose-6 phosphate from the cell, removes the phosphate, facilitating its export from the hepatocyte. And then we have the inverse enzyme glucokinase that takes up the glucose and puts a phosphate on it so that they can go into glycolysis. And you can see that glucose-6 phosphatase is expressed in periportal hepatocytes, whereas glucokinase is expressed in pericentral hepatocytes. And this is interesting. It might mean that the pericentral hepatocytes are feeding on the glucose coming in from the periportal hepatocytes. It also means that it can also be used for responding fast to hypoglycemic challenges. I won't go into this, but it's an interesting system that works sort of in a production line pattern. Another interesting system is glutamine-glutamate cycle. So the enzyme glutaminase takes up glutamine, breaks it into glutamate, and you see it's expressed periportally. And the inverse enzyme that takes glutamate and converts it to glutamine is expressed in the most pericentral layer. Now, this is a really weird, like at first this was really weird to us. Why would a system want to do something like that? Break up glutamine in one zone of the tissue and then build it back in another zone. But when you think about the function of this, it actually makes a lot of sense. This is part of the urea cycle. We know that the body breaks up proteins all the time. When proteins are broken down, ammonia is generated. Ammonia is very toxic, and the liver is responsible in detoxifying ammonia through different enzymes, such as this ASS1. And basically, the detoxification of ammonia through the urea cycle, so ammonia is turned into urea, requires also the breakdown of glutamine into glutamate, okay? So as you can see here, urea cycle enzymes are expressed like glutaminase you saw before in the portal layers, but then the innermost layer doesn't apply the urea cycle. It actually expresses glutamine synthetase. And the reason is that if the entire liver would apply the urea cycle, we would get blood coming into the portal region with certain levels of glutamine and glutamate. Then as blood flows inwards, glutamine levels would go down, glutamate levels would go up. The blood would exit the central vein, go throughout the body, and then go through the liver again. Again, we'll have less glutamine, more glutamate, and we'll completely perturb the amino acid balance in the body. So what the system has done here is that within the pericentral layer, we have hepatocytes that take up all the excess glutamate that has accumulated and builds back the glutamine, so that the concentrations of glutamine and glutamate at the portal node and the central vein are identical. And in the middle, we had quite a few layers that performed efficient ammonia detoxification. Okay, the last system I want to show you here with hepatocytes is a neutral bile acid biosynthesis. So we know that also hepatocytes, the liver, are the only cell types in the body that can get rid of cholesterol. And the way that this works is that blood with cholesterol, cholesterol from the blood enters the hepatocytes, and the hepatocytes take up this cholesterol and convert it through an enzymatic cascade into bile acids that are basically secreted into the bile duct and flow in the opposite direction to the blood flow and exit into the gastrointestinal tract. Okay, so if eventually some of it is recycled back to the liver, but a certain part of it is extruded from the body as feces. So that's the way for the body to get rid of cholesterol. And the bile acid biosynthesis process was known to be very central, but since we had a very high spatial resolution, we noticed that the first two enzymes, CYP7A1 and HSD3B7, were indeed very, very pericentral, but the next enzyme, CYP8B1, actually peaked in the second layer. So it was inhibited in expression in the most pericentral layer and peaked in expression in the second layer. And we're not sure what this means. Potentially, it can mean that sequential hepatocytes along this axis are sort of transferring intermediates and performing this bile acid biosynthesis in sort of a distributed manner. Of course, it was important to validate this, so we performed single molecule FISH for this CYP8B1, and you can see that indeed the expression is inhibited in the most pericentral zone, and then it peaks at the second layer in a very different way than CYP7A1, the first two enzymes are expressed. Okay, so I'll just summarize this first part. I've shown you that we can use landmark genes and single-cell RNA sequencing to reconstruct a global zonation map of the liver. We saw some interesting design principles of tissue organization for the liver, such as the allocation of energetically demanding tasks to the outer layers of the lobule, the more highly oxygenated layers. We saw examples of production lines where sequential enzymes in a metabolic cascade are expressed in sequential lobule layers. We saw examples of spatial recycling, such as the breakdown of glutamine and synthesis of glutamine in different layers. There's other interesting design principles, for example, allocation of transporters with different affinities and capacities that match the availability of their substrates. This is just one cool example. I won't elaborate on this, but glucose enters through the portal node with very high, through the portal vein from the gut with high concentration, and as blood flows inwards, hepatocytes absorb glucose, so the concentration of glucose goes down. And what we see very nicely is that the low affinity, high turnover transporter of glucose that works great for high concentrations of glucose is expressed periportally, whereas when the glucose concentrations are low, the system shifts to the expression of the high affinity transporter GLUT1, from GLUT2 to GLUT1, again, potentially optimizing the overall absorption of glucose by the liver. And if you want to read more about this, we summarize these design principles in this review a couple of years ago. Okay, so this is the first part on hepatocytes. I want to shift gears and tell you just in a few slides on other cell types in the liver that are very interesting for us. So we know the hepatocytes, they take up most of the mass and the volume of the liver, but they're not the only cell types. There's other very important non-parenchymal cells. For example, Kupffer cells and endothelial cells. Endothelial cells make up the blood vessels. And of course, they're naturally going to be very heterogeneous, just because there's veins, there's arteries, there's sinusoids, but they're also static. So they sense the same gradients of morphogens, hormones, oxygen. And so we wondered whether these endothelial cells also have zonated gene expression. Same question could be asked for Kupffer cells. Kupffer cells that reside within the sinusoidal channels are static cells. They phagocyte the microbes that come in from the gut and also dead old blood cells. And we wanted to know whether endothelial cells and Kupffer cells also have zonated gene expression signatures. So the first thing we did is to perform single-cell RNA sequencing. We said we're going to apply the same approach as we did for hepatocytes. So we do single-cell RNA sequencing, and we have the expression of the endothelial cells, the Kupffer cells, T cells, B cells, etc. And then we started to look for landmark genes, for the expression of landmark genes. Not a lot was known on endothelial-specific landmark genes. There were about half a dozen genes that were known to be zonated. And we quickly understood that this is never going to work. Unlike the hepatocytes, spatial reconstruction with a small set of landmark genes is infeasible for non-parenchymal cells, for the non-parenchymal cells we had. And the reason is that we simply didn't have enough RNA per single endothelial cell to infer expression. So a typical hepatocyte has about 700,000 RNA molecules. Single-cell RNA sequencing, you sample only 1%, right? So you get something like 7,000 UMI, 7,000 RNA counts. But a typical endothelial cell has 30,000 RNA molecules, not 700,000, 30,000. So when you sample 1%, you get 300 to 500 RNA molecules, and that's just not enough. Any individual gene would usually be expressed in 0 copies per every endothelial cell, just because of this subsampling. And so we need several dozens of landmark genes, and we simply didn't have any prior knowledge on zonated landmark genes in endothelial cells. So we were stuck here. And then we thought about the following solution. So that's the challenge. Endothelial cells are too small to enable reconstruction with a small set of landmark genes. What we did for hepatocytes very successfully, since in hepatocytes, as you saw before, the landmark genes were so abundant, were so highly expressed. Albumin is expressed in 50,000 copies per cell. You sample 1%, you get 5,000 copies, and then, telling the difference between 5,000 copies in a periportal hepatocyte and 2,000 in the pericentral hepatocyte is much, much easier compared to a gene that's expressed in like 10 copies per cell. You sample 1%, and most of the cells have 0 copies. So that's the challenge. And the way we went about resolving this challenge is through an approach we called paired cell RNA sequencing. So what we do here is we said the following. We already have single-cell RNA sequencing of the hepatocytes, and we have single-cell RNA sequencing of the non-parenchymal cells, including the liver endothelial cells. What we want to do now is suboptimally dissociate the tissue so that we have not a single cell, but get pairs of cells of an hepatocyte and an attached endothelial cell, okay? And then we can sort these pairs of cells into the same 384 well plates. So the pair falls into the well and bursts up. And some of the RNA molecules come from the hepatocyte, and some of the RNA molecules come from the endothelial cells. Now, since I have single-cell RNA sequencing, I know which genes have RNA that only comes from hepatocytes and which genes have RNA that only comes from the endothelial cell. And I can use the hepatocyte genes to determine where the pairs came from, if they came from the central region, the mid-lobular region, or from the portal region, the portal zones. And then I can computationally put together many, many pairs and switch to the endothelial specific genes, and then look for differential gene expression between endothelial genes in pairs that are pericentral, mid-lobular, or portal. Okay, so so that's the idea. Extract liver endothelial specific genes and examine their expression in computationally stratified pairs that were zonally stratified according to the hepatocyte transcriptome of their neighbor. So we hitchhike on the information, on the zonal information in the hepatocyte neighbor. Okay, and then we can pull together these doublets, these pairs, average the expression of the endothelial specific genes, and get the comprehensive zonation maps. So how do we do that? What we do is we use a crappy collagenase D, and we do like a very suboptimal tissue dissociation. And then we go to the fax machine and we sort the big cells, which are the hepatocyte, according to Ford scatter, side scatter. And then we also examine the expression, the fluorescence of CD31 antibody. CD31, and we only sort the CD31 positive hepatocytes. Now, CD31 is not expressed in the hepatocyte. And so this 1.9% of events here, as you can see here in image stream, invariably consists of this large hepatocyte with a very tiny liver endothelial cell that's attached to it. So these are the pairs we want. So we use size to sort for hepatocyte and a cell type specific surface marker to sort for the hepatocyte with an attached liver endothelial cell. And there's a few hundreds of genes that are endothelial specific, And by computationally stratifying the pairs, we resolve the expression of these hundreds of endothelial genes. We also validated it massively with single molecule fish. This is an example. The red dots are RNA molecules of aquaporin-1, sort of a pan-endothelial cell marker. And the green dots are RNA molecules of delta-like 4. This is a gene that our reconstruction predicted would be expressed more highly in periportal endothelial cells. And indeed, you see the periportal endothelial cells have higher expression, more green dots than the pericentral endothelial cells. But we've done this validation for several genes using single molecule fish. So we believe the spatial reconstruction of endothelial cells. And now we can start asking questions about specific endothelial subpopulations at different zones. A very interesting subpopulation consists of the endothelial cells that line the central vein. They express very important molecules such as Wnt and responding 3, which are morphogens that are basically diffusing outwards through the space of Disse and are dictating a lot of the zonated hepatocyte gene expression signatures. And so our spatial reconstruction gave us the complete expression profile of these endothelial cells. Very interestingly, in addition to these canonical Wnt morphogens, we found that pericentral endothelial cells also express DKK3, which is sort of a Wnt inhibitor. And when we did fish, we saw that these are expressed in a subset of endothelial cells in a mutually exclusive subset. So there's some endothelial cells that express Wnt2 and others that express sort of this DKK3. So sort of like a gas break niche that secretes both canonical Wnt morphogens, but also anti-Wnt's. We don't know what's the functional role of this, but it's one of the interesting genes that came up in this spatial reconstruction. And so in general, we're very excited about the paired cell RNA sequencing technique. We think it can be applied for spatial reconstruction of other cell types, such as Kupffer cells, by sorting Kupffer cells and endothelial cells that are attached, or hepatic stellate cells. We also think, and we're working on applying this approach for other tissues. This is just an example of a colorectal tumor with two different carcinoma cell types. There's these blue carcinoma cells which express NT5E, CD73, and the green cells that express LGR5, another gene. And the red dots are RNA molecules of CD3E. This is a T cell marker. So you can see that some T cells are attached to the LGR5 positive carcinoma cells. Other T cells are attached to the NT5E carcinoma cells. And you can think of approaches to dissociate this tumor and sort the CD3E together with the EPCAM or some carcinoma marker and perform paired cell RNA sequencing and identify the differences in expression between the T cells that are adjacent to the blue layer or the green layer. So this is just one application of this technique. Okay, so in the last part of my talk, I want to switch gears and tell you about the human liver, because I know it's a very active research at the NCI. What we wanted to ask is whether there are similar zonation patterns in the human liver in terms of hepatocyte zonation patterns, similar to what we've seen in mice, and whether there are also zonation patterns and spatial heterogeneity in human liver tumors. So we know, of course, I don't have to tell you that the liver is a major site of tumors and metastases, mostly primary liver cancer, which is hepatocellular carcinoma, cholangiocarcinoma, and also hepatomas, but of course also liver metastases, colorectal, lung, breast, pancreatic cancer metastasizes massively into the liver. And we don't really have a complete map of the cell types, the interaction and the spatial distribution of cell types in the liver tumor microenvironment. And this is part of the goal of the project I want to tell you about now. So what we did is to team up with friends from Hadassah Medical Center, Gidi Zamir, Samir Abu Ghazala, and Eli Pikarski, who I saw is going to give a talk at the NCI. You're very lucky, very dear friend and amazing scientist. And this was work done by Hassan Masalha, a postdoc in my lab, and Kiran Bar-Halperin, my staff scientist. And basically, we basically took resected human livers from surgeries, 2 cholangiocarcinoma patients, 3 colorectal liver metastases, and 1 benign stage tumor. And from every liver, we took the normal, sort of the adjacent normal, the adjacent non-malignant liver, and the malignant liver. And for every piece, we divided it into 3 parts. We performed single-cell RNA sequencing on one part. We embedded another piece for laser capture microdissection, which I'll show you in a moment. And the third piece we fixed for single molecule fish. And we reconstructed this atlas of about 8,000 cells. The black cells are cells from the malignant part of the livers. The gray cells are from the non-malignant parts of the liver. And purple stains the carcinoma transcriptome. And so within the tumor, we have the carcinoma cells, but we also have cells of the tumor microenvironment, and we can annotate them. So we have basically endothelial cells, both in the non-malignant liver and the malignant liver. We have myeloid cells. These cells, they're called scar-associated macrophages. We have dendritic cells, tumor monocytes. We have T cells, B cells, and a large population of mesenchymal cells, mainly cancer-associated fibroblasts and pericytes in the malignant liver, and stellate cells and myofibroblasts in the non-malignant liver. And the first feature I want to tell you about this atlas here, the cell atlas, is an interesting feature related to the differences between patients, between samples. If we color this atlas according to the patient from which the cells were sampled, we see a very different picture between the carcinoma cluster and the other tumor microenvironment supporting cells. So for example, in the carcinoma cluster, we see that cells from every patient, so the yellow is one patient, cyan is another patient, magenta is a third patient. We see that every patient has its own cluster, its own gene expression signature. Whereas if you look at the tumor microenvironment, it looks like all the colors are intermingled, right? So the scar-associated macrophages, the endothelial cells, you cannot tell the difference between cells coming from one patient or another. And we can actually quantify that. We can basically reconstruct this atlas based on subsampled patients. So reconstructed based on only one patient or two patients or three patients. This is what you see on the x-axis. And on the y-axis, we quantify the amount of added information that every new patient gives us on this cluster. And you can see that for the carcinoma cluster, the black line, with every new patient, we get more information because every new patient is different. The carcinoma cells of every patient are distinct, but for the supporting cells, these myeloid cells, endothelial cells, cancer-associated fibroblasts, after two patients, we get zero added information. And so there's sort of universal gene expression signatures to the tumor, to the liver tumor microenvironment. Again, even though some of these are cholangiocarcinomas, some of these are colorectal metastases, the microenvironment sort of seems to have a universal convergence on some optimal gene expression signature for the tumor. And since this is the case, we can ask how this microenvironment interacts with the tumor. So we have this purple carcinoma clusters, and we looked for receptors or ligands that are specific to the carcinoma clusters that have matching ligands or receptors from these microenvironment cells, from endothelial cells or mesenchymal cells or myeloid cells or T cells, etc. And we sort of did that for each patient, and we created a sort of a consensus map of ligand receptor interactions that occur in three or more patients. And we see that again, these microenvironmental cells communicate with the cancer through very defined modules. So we have the AGF-MET module where the carcinoma cells express MET and the tumor microenvironment cells express DCN and HGF, the ligands. We have the ERBI module, we have the matrix remodeling module with a lot of integrin expression on the carcinoma cells and matching collagens on the microenvironment. We have non-canonical Wnt, TGF-beta, etc. These maps also have sort of canonical motifs, such as the one we see here. So we often see a situation where both the cancer-associated fibroblasts in yellow and the scar-associated macrophages in red supply a major ligand, such as AGF, that communicates with MET receptor on the tumor cells. And in addition, the cancer-associated fibroblasts supply an accessory molecule that modulates this interaction. Same goes for non-canonical Wnt signaling. Wnt5a expressed by both cancer-associated fibroblasts and myeloid cells, and CTHRC1 expressed by cancer-associated fibroblasts modulates this interaction with the cancer-specific receptor. So this is a map that we believe might pinpoint some, points of vulnerabilities for that could be maybe attacked between the microenvironment and the carcinoma cells. And the last part, I just want to say something about the spatial distribution of these cells. So as in the normal liver, when you look at a tumor, it also has very interesting non-random features. So this is the non-malignant liver on the right, and this is the malignant liver on the left. and you see a necrotic region, a fibrotic region with a lot of matrix and not many cells, the malignant core with many cells, and then we have the malignant border. And we also have these lymphoid organs, tertiary lymphoid organs that are almost like watchtowers that are equally spaced along this boundary. And we wanted to ask how these different populations are distributed along these spatial axes. And we started with the non-malignant liver. We first wanted to know whether we can use landmark genes and take the hepatocytes, the non-malignant hepatocyte, and localize them along the lobule axis. For this, we didn't use fish, we used laser capture microdissection, where we basically tell a laser beam we want to analyze a specific part of a tissue. The laser cuts, goes out of focus, produces a pulse, and then this tissue jumps out of the slide and up into a cap of an Eppendorf tube. This is how it looks like on the cap of an Eppendorf tube. And then we can dissociate the tissue and extract the RNA. And this is a technique we've used many times before, mainly in the gut, to resolve zonation patterns along the villus, epithelial, and stromal axis. You can read about it in these publications. And so in our work, we went for the non-malignant liver. We identified the portal nodes, the central veins. We divided the lobule axis into 6 zones, catapulted every zone into a different tube, performed RNA sequencing in order to get landmark genes. Now I'll tell you just in 2 slides something about the computational challenges in extracting landmark genes from laser capture microdissection. This is different than extracting landmark genes from fish. In fish, we segment hepatocytes around the portal node, around the central vein. So we have digital gene expression in hepatocytes. When we do laser capture microdissection, we get RNA that comes from hepatocytes, but also from non-parenchymal cells. And it could be that in some layers, we have more non-parenchymal cells compared to other layers, and that might potentially confound us. And I'll give you an example. Let's say that we have an hepatocyte-specific gene, like this red gene. If we just take laser capture microdissection and example the expression of this gene, the expression would be higher in layer 2 compared to layer 6. And this is just because in layer 6, we have more Kupffer cells and less hepatocytes. And so we might erroneously think the red gene is a pericentral landmark gene, but of course, within every hepatocyte, the expression is completely identical. And so before we extract landmark genes, what we do is we take our single cell atlas. From the single cell atlas, we extract genes that are specific to hepatocytes. So the x-axis here is the expression in the hepatocytes. Y-axis is the maximum expression in any other cell types. And we only examine the red genes, the genes for which we're sure any RNA we see comes from hepatocytes. And we sort of normalize the expression of these laser capture zones over the hepatocyte-specific genes. And this allowed us to obtain a very clean set of pericentral hepatocyte landmark genes or periportal landmark genes. We use these to computationally identify where every hepatocyte came from along the lobule axis, computationally pull together the hepatocytes from every zone, average their expression, and obtain a human hepatocyte zonation map. I'll just tell you briefly, many of the genes in human and mouse hepatocytes are zonated similarly. Here, green is mouse zonation, blue is human zonation, but some genes are also zonated very differently. And this is interesting. This is just an example of fatty acid biosynthesis. Sort of the thought in the zonation field is that beta oxidation, the breakdown of lipids, is periportal in the liver, whereas the synthesis of lipids is pericentral. And this is definitely true for humans. You can see that the main enzymes in lipid biosynthesis, ACLY, FASN, SREF1, the blue curves are zonated pericentrally, but in mice, actually they're periportally zonated. So in mice, both both the breakdown of lipids and the buildup of lipids is periportally zonated. We can also do the same trick of identifying how different cell types are distributed along the tumor axes. And this is my last slide, really. So we have in the tumor, we have the boundary between the non-malignant liver and the malignant liver. We have the tumor core, which are carcinoma cells surrounded by carcinoma cells. We have the fibrotic zones. We have tumor islands, which are carcinoma cells surrounded by fibrosis, and also the boundary between the tumor and the fibrotic zone. And we can sort of deconvolve our atlas and estimate what fraction of each zone corresponds to each one of the cell types, and some of it makes a lot of sense, like hepatocytes are more abundant at the tumor border and less as we go inwards into the tumor axis. Carcinoma is most abundant in the malignant core, of course. Cancer-associated fibroblasts are more abundant in the fibrotic zone. But also we see interesting, unexpected for me, features of the immune cells. For example, the scar-associated macrophages and tumor T cells are more abundant in the tumor border and the fibrotic zone and less abundant in the sort of the tumor core, in the center of the tumor that's away from the border. Okay, if you want to explore these and other data sets, I highly recommend checking out our resource tab in our group homepage, where you can enter your favorite genes and see the human hepatocyte donation profiles and mouse hepatocyte donation profiles. And you can also browse the single cell atlas of the human liver tumors. And that's it. I want to summarize. I've shown you spatially resolved single cell reconstruction that gives us the global spatial division of labor of hepatocytes. We've talked about paired cell RNA sequencing that can resolve zonation profiles of liver endothelial cells and other non-parenchymal cells. Laser capture micro dissection sequencing that can provide a very large group of landmark genes in an unbiased manner. And a cell atlas of the non-malignant and malignant human liver, showing some patient-independent signatures and interactions between this sort of universal tumor microenvironment and the patient-specific carcinoma cells. I want to thank my lab. The people mainly involved here are Karen Bar-Halperin, my amazing staff scientist who did most of the liver work in the lab, together with Rom Shenhav, a very talented master student who did all the computational analysis for the first two works, and Hassan Masalha, a very talented ex-postdoc in the lab, and of course our collaborator, Gidi Zamir, Samir, Eli, Ido, Maria, and that's it. I'll be happy to take any questions.
It's a terrific talk. Thank you, Dr. Itzkovitz. There are a number of questions. Maybe I will call the name and you can unmute yourself and ask the questions by yourself. So the first one is from Dr. Scott. Do you want to unmute yourself and ask the questions?
Yeah, great. Thanks so much for this talk. This is amazing. So I was wondering, and guess this is probably the million-dollar question, is like, how is this zonation being regulated, this gene expression across the zones being regulated? Do you have any ideas from the work that you've done? And particularly, I was really interested in what you showed between the L-sex, just adjacent cells have very different gene expressions. So have you got any ideas on how this is being managed by the liver?
Yeah, it's a great question. Of course, once you have a global map, you can start asking these kind of questions because we know which genes are putative Wnt targets and KRAS and RAS targets and the hypoxia-related genes. And we've done this analysis. Overall, about a third of the hepatocyte-donated genes can be explained by Wnt. So Wnt-activated genes are pericentral, Wnt-inhibited genes are periportal. And the main Wnt niche consists of endothelial, pericentral endothelial cells, but also pericentral hepatic stellate cells. How are they regulated? And of course, so this is that sort of and I want to say there's two pictures we can have in mind. One is that, the liver consists of hepatocytes, which are a homogeneous cell type. So a periportal hepatocyte and a pericentral hepatocyte are exactly the same cell type, and they just sense different gradients of Wnt, oxygen, and then that's why they respond different transcriptionally. A second, more interesting hypothesis is that these are actually different cell types. Okay, so if you pluck out the periportal hepatocyte, you put it around the central vein, the question is whether it's going to behave, it's going to sense the same microenvironment, is it going to give the same expression signature as its neighboring pericentral hepatocyte? And this is something we're very interested in studying massively. I can tell you that we've done an epigenetic map of the liver lobule, and DNA methylation patterns of hepatocytes in the portal and central regions are different. For many genes, many of them are also correlated. So genes that are highly expressed around the central vein tend to have DNA methylated in the central vein and not in the portal, around the portal vein. And so we tend to favor that these are also, developmentally different cell types. Of course, we know they're very plastic. We know that following regeneration, they can, like periportal hepatocytes can reach the central vein and reprogram. But I think it's going to be a combination of these two. It's like the gradients of morphogens, oxygen, nutrients, hormones, Wnt #1, no doubt. Then afterwards, glucagon, some hormone, pancreatic hormones, but also the internal epigenetic zonation, which I think also plays a role.
Great. The second question, I think, is from Chi. Would you like to ask about yourself?
Okay, thank you. So it's really nice work. I'm just curious, so do you think the hepatocyte will keep the genetic features during regeneration? Because during liver regeneration, like the hepatocyte will experience like some differences or trans differences or something. Really curious about that. Thank you.
Yes, it's a great question. One of the most exciting projects in the lab at the moment is regeneration after zonal damage. What we do is acetaminophen intoxication. So we give an acute dose of APAP. This kills all the pericentral hepatocytes. So sort of like the 3-4 inner layers are necrotizing. But then within 4 days, hepatocytes, periportal hepatocytes and mid-lobular hepatocytes start dividing, pushing inwards the hepatocytes. And so a mid-lobular hepatocyte is pushed inwards and becomes a pericentral hepatocyte. And amazingly, gene expression goes back to normal. It's a very, very interesting time frame. There's a lot going on here. There's monocytes coming in, eating the dead hepatocytes. There's hepatic stellate cells that are activated in a spatially inhomogeneous manner and then are regressing. And we're very interested in this. It's a reprogramming event. So they don't. What's very interesting is that they don't just shut down portal programs and upregulate central programs. They revert to an embryonic state, a fetal state. So they erase their identity, upregulate, alpha fetoprotein, different like genes that are not expressed at all in hepatocyte, but were expressed in the fetal stage, and then approach probably the Wnt niche and take on the new identity. So I think it's a fascinating. There's two types of regeneration. There's like the 2/3 partial hepatectomy, which is like it's a regeneration, but it's sort of regeneration that preserves the lobule structure. You just get more lobules or bigger lobules. But there's also these zonal regenerations, which I think are the best spotlight to really understand the plasticity of hepatocytes and the zonal plasticity of hepatocytes. So definitely this is something we're studying at the moment.
Thank you. And the next question is from Dr. Shin.
Oh, hi, Shalev. This is a great work. So I have two general questions. So have you ever done any spatial omics in some injured state, like or in any other kind of disease, like in insulin resistance, like mouse models or NASH or fibrosis? And another question is, so have you ever considered any circadian rhythm into your context?
Yeah, I'll start with the second one. So we have a new paper with Felix Neff, which is basically a spatial chronobiology, it's a space-time logic. We did the exact same study I showed you at the first part of my talk, but around the clock, like every 6 hours, we killed 2 mice, extracted hepatocytes. So we have 20,000 hepatocytes throughout the day, reconstructed space-time. And, the question was, we know that 30% of the hepatocyte genes are changing along a circadian rhythm, and we wanted to know whether all of the zonation profiles sort of are pushed up and down, or whether there's going to be like sub-lobular circadian oscillations. Maybe, some genes will switch zonation patterns. So this is something we've studied. We see that the general shape of the zonation profiles remain intact, and sort of circadian rhythm is sort of a multiplicative factor that modulates the levels, but usually doesn't change the ratios between portal and central hepatocytes. Injury models, so we were doing this APAP regeneration and some models of biliary diseases. We're mainly interested in applying our paired cell approach because this has the power to really identify interactions between activated fibroblasts and immune cells, cholangiocytes and fibroblasts, cholangiocytes and immune cells. So we really want to explore this universe using the paired cell approach. I think there's a lot to be learned here that we don't know yet.
Great. So the next question is from my mentor, Dr. Xinwei Wang.
Oh, hi. Yeah, it's fascinating talk, fascinating science. I want to say that actually during certain points that at least 170 people attended for this session. Very, very nice. Thank you. Well, so my question is about the sexual dimorphic organs. You know, liver is obviously one of those organs. I wonder whether there is some sort of a the pattern, zonation pattern in male-female difference, but we know that there's a huge difference between male and female in terms of hepatocarcinogenesis.
It's super interesting. I know there are differences. There's a paper that has done actually laser capture microdissection of central portal in male, female, and highlighted some differences. We haven't looked at that. I think it's very interesting. And again, with the new approaches, these single cell atlases are coming now. You know, there's more and more single cell atlases with normal hepatocytes, some from male, some from female. We have the landmark genes. You know, most of the landmark genes would be universal. So we can, we could sort of computationally infer the position of the hepatocytes and start looking at the, zonally stratified differences between male and female. I think it's very, very interesting. We haven't done that.
Thank you. And the next question is from Scaver. He has some kind of issues with the microphone, so I will help him to ask this question. Tetraploid hepatocytes are common even in normal liver. Do you distinguish between diploid and tetraploid hepatocytes in single molecule fish analysis?
Yes, we've been fascinated by liver polyploid, the hepatocyte polyploid, and been studying it massively in recent years. These tetraploid hepatocytes, they have twice the transcriptome of a diploid hepatocyte, but they also have twice the volume. And so the null hypothesis is that, if every DNA copy of a gene shoots, transcribes in the same rate, and RNA degradation between tetraploid and diploid is the same, then in terms of the concentration of RNA, it's going to be the same. You're going to have twice the amount of RNA made spread over twice the volume. But then you can think of situations when you might have ploidy-specific gene expression differences, like proteins that operate on the membrane. So if a cell has twice the volume, it has less than twice the surface area. So maybe there's going to be dosage compensation in polyploid. So we've studied that massively. There are several papers that claim there was no differences in the concentration of RNA between diploid and polyploid cells. And this is basically our conclusion so far as well. I can tell you that after regeneration, after APAP, after these 4 days I told you about, where gene expression is zonation is back to normal after 4 days. But amazingly, the new volume of the hepatocytes in the pericentral region doesn't come from more cells, it comes from larger cells. 1 week before, all the pericentral hepatocytes are tetraploid. 1 week after regeneration, all of them are octoploid or 16N. But gene expression is almost exactly the same. So what is the role of polyploidy? I don't know. It could be sort of backup copies, especially in this mid-lobule zone where you have a lot of hypoxia and xenobiotic metabolism, reactive oxygen species. So maybe you have more backup copies, the hepatocyte might be more viable. But yeah, in terms of average gene expression, looks very, very similar, surprisingly to me.
Great, and there are still a number of questions, but I think because of the time limitation, I can only choose one from here. So the next is Dr. Paulat Shilian, and would you like to ask by yourself?
Yes, thank you. Hi, Shalev. Fantastic presentation on a topic that close to my heart. So you mentioned that fatty acid oxidation and lipogenesis in mice is not zonated. So is it at least at the RNA level?
No, it is zonated, but both of them are zonated periportally.
Periportal.
So fatty beta oxidation, the breakdown of lipids is periportal. That makes a lot of sense because like sort of the classic idea in zonation, you break up lipids periportally to get ATP. You burn the lipids, you get ATP, and then you synthesize them pericentrally. In mice, that doesn't work like that. You both break down lipids, but you also synthesize lipids in the same zone, in the periportal zone. In human, it is inversely zonated.
Yeah, is it possible that in mice the it's still zonated functionally, but it's not at the RNA level, maybe at the protein level translation
Yes.
or post-translational modifications? And also, would you elaborate a little bit on the correlation between the zonation at the RNA level and the protein level?
Right. So we have a paper on Nature Metabolism on something we call spatial sorting. What we basically do is we create omics maps of the tissue, proteomics map, microRNA map. So what we do is we take our zonation profiles and we identify zonated surface markers, and then we dissociate hepatocyte and basically sort them according to the zonated surface markers. So we sort hundreds of thousands of hepatocytes from different zones that are stratified by the zonated surface markers. And we found, and then we can measure both RNA and proteins. We found that the zonation of the proteins overlaps almost perfectly the zonation of the RNA in the liver. By the way, it's not like that in the gut. Different in the gut, there's anticoagulation, there's interesting stuff going on. But in terms of translational control, I think that's secondary. Post-translational regulation might be, there might be something going on there, definitely.
Okay, great, thank you.
Thanks.
We would like to thank Dr. Itzkovitz again for the fantastic talk. And Anu, do you have anything for announcement?
No, just thanks again to Dr. Itzkovitz for a really fascinating presentation, really great science. And thanks to our audience for joining. And I hope you can join us again on Monday, June 21st for our next seminar in this series. So thanks again.